My name's Lockie McBurney. I'm the Biodiversity Officer at Nillumbik Shire Council, and helping me tonight present is Beth Hamilton. She's an Environmental Projects Officer with Council as well. So tonight we've got an amazing guest speaker in Dr. Kevin Rowe from the museum. He's the curator of mammals there and has been since 2011, I think, Kevin, but you'll be able to correct me on that if we get there.
That's
So
correct.
I'd like to start off tonight with the acknowledgement of country. So we acknowledge the Wurundjeri Woiwurrung people as the traditional owners of the country on which Nillumbik is located. We pay our respects to elders past, present and future, and extend that respect to all First Nations people. We respect the enduring strength of the Wurundjeri Woiwurrung and acknowledge their sovereignty was never ceded. So tonight we're going to be listening to Kevin in as much as we can, explain as much as he knows about particularly rodents in Australia and some other native mammals, but he's going to cram a hell of a lot of knowledge into a very fast-paced hour, I would think, based on his experience and his CV. It's going to be great. So this is one of our environmental events where we're trying to get experts in to present on various subjects. So this will be the first in a series of people coming into and chatting to us like this. So if there's other experts that you want to hear from, let us know here at Council and we'll see if we can facilitate a really cool presenter from someone with a fascinating job and job title. So that's enough from me. I'm going to throw over to Dr. Kevin Rowe to start his presentation. So if you want to use the chat function and Q&A as we go, we'll answer questions at the end if we've got time. So over to you, Kevin.
Great, thanks Lockie, and thanks Beth for organizing this, and thanks everyone for coming to hear me talk about rats on World Rat Day 2025. Before I begin, I'm also a Nillumbik resident, and it's my pleasure as an American immigrant to Australia and a student of diversity to acknowledge the Wurundjeri people of Greater Melbourne, on whose lands I have both the privilege to work, live, and raise my family. So I offer my respects to their elders past, present, and emerging, to any First Peoples in the audience today, and to the many Indigenous peoples who have welcomed me onto their ancestral lands around the world. So today, as we know, is World Rat Day.
Kevin, so
yeah.
you've got the slide on. There's always something in a webinar, but we can see your next slide. View? So you're not on. Yeah, I don't think you're in presenting mode.
Okay, I will. You can see the full presenter mode. How is that now? Is that?
Yeah, that's the one. Yep.
Okay, much better.
Thank you.
Not that there's a ton of notes here, but it'll be confusing to see the next slide. So my chance to talk to you about rats and the coolness of their diversity, particularly our native Australian rodents. And in the middle here is a little icon I borrowed from our Year of the Rat 2020, a year that everyone remembers very fondly. But we kicked it off with the Year of the Rat and gave a talk on rats then. So as Lockie said, I work for Museums Victoria. I work in the Natural History Collections. I work in the Sciences Department and Museums Victoria's Research Institute. Collections globally house a vast record of biodiversity on our planet. They house the type specimens upon which nearly 2 million scientific names are based, and they document centuries of global change. They include extinct species like the thylacine and the Gould mouse on the lower left that I'll talk about later. And they include many different preparations of specimens, from skins and skeletal remains and fluid preserved specimens, and increasingly cryopreserved specimens. They also include many digital records of specimens. Increasingly, we're collecting many new camera records and acoustic records that become part of our collections and digital collections. Across Australia, around nine museums contain more than 300,000 specimens collected from across Australia and adjacent regions. And here's a map of where those collections are placed, are from. And the Australian collections work in a global network of museums with more than 10 million mammalian specimens preserved in collections over the last 200 years, really, spanning the last 3 centuries, from the 19th century to now the 21st century. So today we're really talking about rats, and that's kind of a confusing term sometimes, because what is a rat? World Rat Day is billed as a day to celebrate pet rats or fancy rats, but to many people, rats are pests, not pets, like these black rats, or
do for a couple of 100,000 carpet prices. Ever seen mice like this?
So mice, are they rats? That's an open question here, but I would say mice are really small rats and rats are really big mice. There's really no difference between them, and I'll talk about a little bit that later, but today I'm going to really lump mice and rats into one group, and they sort of elicit the same response, especially when Steve Irwin here shows you these mouse plagues and this sense of pests. But what about this animal? Is this a rat? This is a cloud rat from the Philippines. They live in the high canopies of Philippine rainforests, and they grow to a size of up to 2 kilograms. So rats are rodents. Hopefully, we're loading that. Rats are rodents. Hopefully that's fairly obvious to everyone. And I'll talk a little about where rodents fit into the mammalian tree of life. So this is a slide showing a dot for each of the 6,759 mammalian species. Actually, there's a few less dots because I made this slide a few years ago, and every year we're adding around 50 to 100 new mammalian species to the list. Since 2005, which was the last edition of the Mammal Species of the World, we've added over 1,400 species of mammals to the mammalian species list. So each of these dots represents a species. As I said, this dot here represents all humans, all human diversity, and from all our diverse backgrounds and walks of life. So we break mammals into orders, and we recognize 27 orders, one of those being rodents, Rodentia, and as many of you probably know, Rodentia are by far the most diverse, representing about 40% of mammals. They're matched. In second are the bats, with about 20% of mammals, and then the remaining 25 orders. And so this is sort of laid out in the order of their diversity. So rodents have almost 2,700 species. Based on species we know or yet to be described, there's certainly more than 2,700 species of rodents. There's probably over 3,000. So what is a rodent? The word rodent comes from the Latin word rodere, which means to gnaw, and the thing that makes rodents exceptional and exceptionally successful is their gnawing toolkit. So they have these incisors in front that have an iron-rich enamel on the outer surface that wears slower than the inner surface, which is soft dentine, and so they have these ever-growing and ever-sharpening chisel-like front teeth. They have a gap that we call a diastema between those teeth and a bank of chewing molars. And those allow them to access food resources that most animals of their size could never access, animals much bigger than them. So they can gnaw through many different things. And then because they can separate the gnawing from the chewing, they can process food in really effective ways. And it's an extremely successful toolkit. Not all rats fit into this toolkit. This is a species that we described in 2012. It was known to the Mamasan people of central Sulawesi as Sapiu, but completely unknown to science. It's a worm-eating rat that we described as Posidentames vermidex, Posidentames meaning few teeth. And the reason we call it few teeth is because it has absolutely no molars. It's the only rodent among 2,700 species that has no molars. And it has these upper incisors that cannot gnaw. That little fanged-like tooth is a single tooth with two cusps that it uses to snip up earthworms into bite-sized or swallowable little pieces. So it's a rodent, it is a rat, but it's completely departed from the toolkit that's made rodents so successful. So one of the exceptions that proves the rule. So are all rodents rats? The answer to that is, as we wait, there we go. No, not all rodents are rats, but more than half of them are. And the thing is that things we call rats or mice evolved many times within rodents. So the two most substantial families are the Muridae, which are distributed throughout the Eastern Hemisphere, sometimes called the Old World, and then the Chrysetidae with large radiations in the Americas. Every time we get rats evolving within rodents, there are also mice, because rats are just slightly bigger things that we call rats, and mice are slightly smaller things that we call mice. So rats and mice are not a single thing, a single group within an evolutionary context. They've evolved multiple times within rodents. So does Australia have native rats? Bit of a rhetorical question. Of course they do, but there are about 65 species in 14 genera in Australia. They're part of two much larger radiations centered on the Sahulian continent, and particularly in the island of New Guinea to the north. So there's around 180 species in 34 genera that emerged from the radiations of rats, of rodents, on the Australian continent. So that begs the question of, how did Australia's rats get here? Well, more than 100 years of work with collections, examining specimens, comparing them, sort of classic taxonomy and systematics, describing species, finally resolved that all Australian rats are members of the family Muridae and in the subfamily Murinae, a group with a distribution throughout the Eastern Hemisphere from Africa through Europe, Asia, the Philippines, and into Australia. It's the most diverse subfamily. It's also the most diverse family, Muridae, but the subfamily Murinae has over 700 species, and the Old World rats and mice comprise about 1 in 8 mammal species on Earth, just this one radiation. They include the lab rat and the lab mouse, also the pest species, the mouse and sewer rat, the brown rat, and the black rat, but includes the radiation of all these other species, most of which are not pest species or used in laboratory studies. Surprisingly, this extremely diverse family, the most diverse family of mammals, is relatively young, with an origin about 14 million years ago, somewhere in southern Asia, Southeast Asia, with a distinct fossil record from the Siwalik Hills, which are on the border of Pakistan and India. And 14 million years might seem like a long time, but it's really 50 million years after the KT boundary, when dinosaurs went extinct and most orders of mammals diverged from each other, and most diversification of mammals happened. And rodents have an origin going back to that time, to the KT boundary, whereas rats and mice, Old World rats and mice, Eastern Hemisphere rats and mice, really only have a much younger origin, but are spectacularly diverse. So today in collections, we also examine traditional specimens, but we rely increasingly on tissues and DNA to understand the origins of diversity and its evolution. So that involves DNA analysis, which we can do in our labs at Melbourne Museum. And we use DNA then to construct phylogenetic trees, similar to a family tree. The way we can infer relationships among people within a family, we can infer the relationships among species through time, as shown with this tree of life for the great apes. So this is a tree of the Murinae. It's reasonably complete with almost all genera and about 70% of species. The other way we can present trees as a circle. So those last trees were flat, and you can imagine these have been, it's just been rolled around so that we can fit more species in it. Every species is a tip on the tree here, and then the relationships among species are what we call nodes inside the tree. You don't have to understand all of that, but one of the key things about trees is that the farther towards the center you are, the older, the farther back in time you are. So imagine that the very center of that tree is 14 million years ago, with the origin of rodents, or murines, and then younger is the present day. And what we can do is map the distributions of these species onto the tree and infer where their origins lie. And you can see the Australian radiations, including New Guinea, are shown in green here. And the oldest And the most diverse one is a tribe called the Hydromyini, and you can see they're nested within this darker blue color, which comes from the archipelago we know as Wallacea, which is dominated by the island of Sulawesi in Indonesia, and so that they have a center of origin through that archipelago. And deeper into the tree, the origin, as we expect from the fossil record, is in continental Asia, which expands from Pakistan, India, through to Java and Borneo. There's a second group of native Australian rodents here in the Ratini tribe. They include the genus Rattus. And again, you can see they're much shallower in the tree, so they're much younger. And they're also nested within a group coming from the Indonesian archipelago before reaching the Australian continent. So what do we know from this tree? We can end from our basic understanding of murine relationships. We know that murines crossed from Asia all the way to New Guinea twice. First, they crossed 7 or more times into Wallacea, and 2 of those crossings spawned radiations in New Guinea. And then from New Guinea, rats colonized Australia. That spawned quite a bit of more complicated biogeographic events, with New Guinea populating back into Wallacea, Sulawesi sending lineages back to the Asian continental shelf, colonizing the Philippines, and then repeated transitions between New Guinea and Australia. So to summarize that a little bit and to give you a temporal context, around 6 to 8 million years ago, a progenitor rat floated across open ocean to New Guinea, the proto-island of New Guinea. New Guinea at that time was a complex of multiple small islands, and then diversified within that island archipelago. And then it wasn't until another 2 or 3 million years that rats finally colonized to Australia, the mainland Australia, and they're diversified into around 5 genera of rats alive today. They did this at least 4 more times from New Guinea into Australia. And then about 1 million years ago, a second colonist from Sulawesi reached New Guinea and then quickly spread to Australia, giving rise to our native Rattus. So who are these Australian rats? And I'll start this in a phylogenetic framework. So first, there's the earliest, sometimes what used to be called the old endemics, the Hydromyini, that arrived into New Guinea first and into Australia first. And I'll now give you a slide for each of those species, if you bear with me for 1 minute. So then a second colonization by rats, the Ratini, gave rise to 7 species in Australia. And some of you may have noticed some golden titles going past, and those are our 3 native rats of Nillumbik. Unfortunately, that's not an enormous amount of diversity considering how many rats there are, but they are native rats. So we have 1 Hydromyine, the Racali, the water rat, Hydromys chrysogaster, and 2 of the native Rattus, the bush rat and the swamp rat. So that diversity was living species. It doesn't include 14 Australian rats that have gone extinct since European colonization. That's 22% of species that were alive at the time. So it's a disproportionate number of extinctions, even for the number of mammalian extinctions in Australia. So this was even more diverse than it is currently. And I'll speak to just one of these extinct species in a moment to give you a sense of what we can learn from museum collections. So this is Pseudomys gouldi, the Gould's mouse. It was named after the eminent British naturalist John Gould, and the specimen was first collected by scientists on the HMS Beagle in 1837. It actually wasn't collected by Darwin. Darwin was taking a nap that day, but other naturalists on board the HMS collected the first specimens that went back to the Natural History Museum in London and formed the scientific description of the species. It then was thought to be last seen in 1857 when Wilhelm Blandowski, the first curator of zoology at Museums Victoria, collected this specimen on his famous expedition into the arid interior. One of the things that's remarkable about collections work today is that we're able to get DNA out of these very historic specimens. So imagine this Blandowski specimen is more than 100 years before Watson, Crick, and Franklin discovered the structure of DNA, and yet we can sequence genomic material out of the specimen by taking a very small little 2 millimeter slice off its foot. And so that's what we did in this study. This was by my former PhD student, Emily Roycroft, who's now a faculty at Monash University. Just exceptional work done by her. And this phylogeny includes 40 living species and 8 extinct species of rodents. And when we put Gouldi into this tree, we discovered that it was basically indistinguishable from a living species, the Shark Bay mouse, Pseudomys fieldi, and the difference was less than variation we see within most species. And so we removed Gouldi from the extinct list and resurrected the name in the living population, which survived on a single island in the Shark Bay on Bernier Island. And so what we had discovered is not just we'd not just removed an animal from the extinction list, but shown a continental collapse of a species. So a species was distributed from New South Wales through Victoria all the way across the coast, surviving only on a single island. And that was evident in the genetic diversity that we could get from these specimens. So this is sequencing across the genome, many millions of bases, 10,000 protein coding genes. We can estimate the genetic diversity or the heterozygosity. And what we found is that the extinct species, including Pseudomys gouldi, these last specimens, had genetic diversity comparable to large continentally distributed species. So what was collapsing were huge, widespread, very abundant species down to extinction. In the case of Gould's mouse, it survived on the single island, but now persists with lower genetic diversity than any other living rodent. So this question then, are Australia's rats threatened, is probably obvious. Yes, of course they are. About 20% of species, living species, are threatened with extinction. And we've, as I said, lost 13 species to extinction. One of them, Gould's mouse, not being considered extinct anymore, but quite a collapse. So just to talk about this, I'll give you a little vignette. I'll end with a vignette on one species, the smoky mouse, Pseudomys fumius, EPBC listed endangered species. And here's a lovely photo of one. Smoky mouse records are sparse, but since its original scientific discovery in the Otways in 1933, it was only ever recorded from a handful of areas of southeastern Australia in the 20th century. It's endemic to this small part of the world, but its distribution is far from widespread and continuous. In the 21st century, smoky mice have disappeared from several areas, including the Otways, where their type locality is. And the population and also in far eastern Victoria, and then the populations in the Grampians are isolated from the other populations by around 350 kilometers. And we know they're genetically distinct and quite a different population. In the Grampians, they persist in small patches, spread over approximately 6 kilometers along the 2 mountain ranges, the Victoria Range in the west and the Mount William Range in the east. And they're isolated by about 25 kilometers of unoccupied habitat. And genetic analyses show us that they haven't really been moving between the two mountains at all, exchanging any genes for quite some time. These records reflect over a decade of surveys from 2012 to 2024, and with the white crosses representing places where we never found them and orange crosses where we did. So we've been monitoring the population for quite a while since then and trying to track where it is, leading to deploying of permanent cameras that were active that are in the Victoria Range and in the Mount William Range up until this year. And then, of course, in December 2024, a fire erupted in the southern part of the Mount William Range and expanded to burn 76,000 hectares into January 2025, threatening Hall's Gap and other homes. Fortunately, it didn't burn Hall's Gap, but it burned all of known Smoky Mouse habitat. Immediately after that, just as we were preparing to respond to that fire, the Victoria Range fire and Wallaby Rocks fires merged to form this another giant fire, burning almost 60,000 hectares, again burning all of our known sites. This is what Mount William looked like when we were first got onto the site on the 14th of February. Here we're looking south towards the Mitchell Plateau. The Mitchell Plateau being one little island of green that didn't burn and may hold a refuge for Smoky Mouse in this environment. Our cameras that were deployed looked like this one on the left, completely scorched down to the batteries. And so we've spent the last month and a half replacing and now revisiting those cameras. And above the camera, you see an acoustic device where we're trying to look at how bird communities are responding to the fire as well. So from Mount William, looking across, the fire burned through Fyans Creek, through the Sarah Range in the middle of the park, and then the Wallaby Rocks and Victoria Range fires burned all of the Victoria Range 25 kilometers to the east, the other stronghold of the Smoky Mouse. This is what the Victoria Range looked like. This is one of our core Smoky Mouse sites. This is the first site where I trapped Smoky Mice back in 2012, and we've been monitoring this site really ever since. This is what it looked like last year before the fires. So it was very lush, green, full of lots of vegetation. But this is what it looked like in 2013 at the Victoria Valley fire. Almost a worse fire. You can see that there's a lot more standing dead vegetation in the 2025 photo than the Victoria Valley fire of 2013. So we've been here before. The difference this time is that we have information. So we had two cameras in this gully that are the only two cameras that didn't burn throughout our 35 cameras that were deployed through the park. So those cameras captured the fire burning through on the 29th of January. And they captured Smoky Mice surviving in situ from the 31st onward. And I was just in the Grampians again, checking our cameras earlier this week on Monday. And here's the most recent record of that. So still at the end of March, we still have Smoky Mice on site in this core site. We also have foxes and cats. On the right is a video that I won't show of a cat actually killing a Smoky Mouse on the site on the 22nd of March. And we've been monitoring this site with basically permanent cameras since 2019, and rarely have we seen so many cats. So there are at least 3 cats on the site right now walking through and going past the stations right where our Smoky Mice are persisting. So there are major threats to the Smoky Mice that persist. And if you want to hear more about the details of that, we have produced a video. You can find it on our Museums Victoria YouTube page. There's quite a few other really excellent videos there as well. We have a second video about to drop. So the first video is a little bit depressing, a first look after the fire and my emotional response to seeing a beautiful place that I know very well scorched. And then the second one has a bit more of the hope of Smoky Mice persisting, and there will be more videos to come on how the populations recover. So that's really all I have to say today. So thank you and happy World Rat Day.
Thanks so much. That was awesome. I learned heaps. So I've got a ton of questions. If other people have questions as well, make sure you share them through. Just quickly before I jump into some of the questions, I was at a conference recently where the keynote speaker is Nick Porch. Dr. Nick Porch is he's insect fanatic, and he was talking about biodiversity being represented by insects, but they get none of the funding and none of the attention. So is there anything you want to just give you a free rein to have a chat about it and plug research into small mammals, in particular rodents?
Yeah, I mean, I think research as a whole is facing a number of threats globally right now. And with conservation, one of the challenges is that we want action. And so we want to fund action. And I think that's reasonable. But sometimes we don't even have the basic information to know what actions to take or where to take the actions. And that's been the smoky mouse model for me. What do you do for a species where we didn't know where they were, how many there were, how their population was changing? And we spent the last 13 years trying to build a better record of smoky mice and have a more rapid response to catastrophic events like fires. It's going to be fires and droughts that are catastrophic, and then the feral predators on the ground are going to be the final assassins. So yes, there's definitely a need for more information on biodiversity. We don't even know the number of species in Australia, for instance. So the Australian Academy of Sciences produced a decadal plan for taxonomy, the describing of species, and that estimates that we have a scientific name for around 25% of species in Australia. So there's a lot of research to even catalog what's out there. Entomology insects are probably the greatest biodiversity collapse and catastrophe we're facing right now that is going mostly unrecognized. We're losing insects so rapid we're not even aware. We're going to lose species before they're even named. And insects are really the cornerstone of ecosystems and really central to the survival of smoky mice and many other mammals and birds and other animals that we're familiar with.
Absolutely. And, cheers for all the hard work before the fires as well. Doing all that monitoring work helps inform the response afterwards. So, without institutions like yours doing that amazing work pre-fire and tracking all of our monitoring, then we can't respond in the same way that you have and learn from these huge impacts as they happen. So, cheers for that one. We got a question from. Oh, do you want to do it, Beth?
No, you got it. You don't need me.
So Chris Coburn asked if is there fox and cat control being done since the fires? And he also added, do the lures in front of the cameras make the mice vulnerable to cats, etc., when the mice are distracted trying to get food from the lure?
So the first question is a great one. Absolutely, yes. So we work closely with the Grampians ARC team, who are responsible for predator control. And they fortunately got additional funding to respond to the Grampians fire, and they are responding to the sites where we say smoky mice and cats are. And we were on the ground with them at the beginning of March, and then when I was back this week, there were already baits in the core site where we had detected foxes. Cats are very difficult to control, but they're doing their best efforts with the funding and resources they have, but they could definitely use more resources to control, especially as the population is trying to recover. We know these key assets are there. Smoky mice are still there, and whether they will persist or not depends on whether they're consumed by cats and foxes. Because we think that for smoky mice, there'd be plenty of food on the ground for them. As to the second question about lures, I don't think so. So we've surveyed about 1,600 sites across the state with these baited lures, and we've rarely ever detected a predation event on them. This was an exception for us. Typically, when we're monitoring smoky mice, when we're doing long-term monitoring, we have no baits or lures. We have them completely unbaited, and we're able to detect them because we don't want to bias our detections. Right now, we're just trying to confirm they're there. So I don't think the lures are doing any more to increase the probability of cats killing them, but we don't have hard quantitative evidence to know other, one way or the other. In my experience, watching videos of smoky mice and other animals, they're not getting too distracted with the bait. They're kind of jumping around and running past it, and probably that's probably not what's getting them killed. And the one video of the cat killing them, it wasn't like the smoky mouse was at the bait station and the cat pounced on it. The cat carried the smoky mouse into view on the camera. So that's my anecdotal evidence about. I don't think that bait tubes or collecting data on smoky mice is increasing their deaths to cats. Cats are finding them and killing them, and we're not even noticing it.
Yeah, it was amazing seeing the first footage that you showed with the smoky mice surviving, and you brought us up on a high, and then you smashed us down with the next photo with the cabinet straight away. It's pretty disastrous. We got another question from Sarah and friends about our native bush rats are Rattus. Also, the European rats are Rattus. You said earlier that the Australian rats are more recently evolved. Is the evolution within the same genus, or are they still very similar, or maybe just named similar? From Tim.
It's a fantastic question, Tim. So you're right. The introduced European rats are also Rattus, and our Australian Rattus are also in that genus for now. So I can tell you that the two diverged about 3 million years ago, somewhere in Southeast Asia, and the Australian native Rattus and New Guinea native Rattus are more recent origins. They came from Wallacea, probably from Sulawesi to New Guinea about 1 million years ago, and then spread and diversified into around 30 species in Australia and New Guinea, with 7 species native in Australia. But interestingly, their relatives, which are also on Sulawesi, so all those animals diverged from the other Rattus that include the European rats about 3 million years ago. And we actually think that there is an available different genus name for them that we might elevate. And we're collecting a bunch of new genomic data now from across the family Muridae. We've sequenced about 800 species in that. And with that, we're going to try to resolve some of these generic confusions. Rattus is one of the classic messed up genera. There are species in it that aren't even closely related. There's a Rattus from the Philippines, a Rattus from East Timor that aren't Rattus. There are Rattus from Christmas Island, spoiler alert, that are not Rattus, and in phylogenetic sense. So the problem is that Rattus is a very difficult genus to work on from morphology, and there's a lot of diversity in that genus, and it requires a lot of investment to resolve. But they are about 3 million years divergent from the European Rattus. So if that's similar to you, then yes. But the Rattus that live in Australia are very different ecologically. They do all kinds of different things. They live in the central deserts. They live in, like bush rats, in our forests, or swamp rats in more open habitats, except in Tasmania, where they also live in forests, or except in the Grampians, where we have swamp rats but not bush rats, and the swamp rats live in the forest. So they're actually quite diverse in their lifestyles and ecologies, and doing very different things. And the European Rattus will live in your homes, but most of the native Rattus will avoid your homes, except unless you live in a big bush block, you might get bush rats running around your porch.
It's a great
question.
If
you've got like rats that are in and around the house in suburbia, the likelihood is that they're not going to be native rats.
Yeah, very unlikely. My most common question at the museum, because we have a public question, if you ever have questions about rats, you can send it to ask us@museum.vic.gov.au, and we get those questions. Is this a native rat? And 99% of the time, it's a black rat. So, but I mean, in Nillumbik, we have very many big bush blocks. So you might have a bush rat in your bush block around your house. So don't go willy-nilly killing them.
Another Nillumbik-centered question from me. I'll share this one. I know this isn't, if everyone can see that. This is just a screen from iNaturalist showing Ricali records from in and around our area. And there's only a handful of records, and most of them look like sort of roadkill or dead finds. So how widespread do you think Ricali would be through our, two pretty major river corridors there in the Diamond Creek and the Yarra? Is it just the lack of work being done on them? Are they just too reclusive? Do people just ignore them when they see something scurrying around? Is it because there's not enough work being done? What were our chances of seeing Ricali in part of our shire?
Well, yeah, I've never seen them walking along the Plenty River, where we walk a lot because we live in Apollo Parkways. And I think it's a mix of all those things that I think the rivers are degrading in a way that's not suitable for them. I think there isn't much work on surveying them, and we don't have a good handle of how many there are, where they are. But they're relatively easy to identify, swimming on the surface. They have that long white tail tip. If you find one dead, do report them. I think they're getting underreported. Our sense is that they'll occupy almost any water body. You can see them down in the bay off St. Kilda. If you go down to where the penguin colony is there, you can see Ricali swimming around. So they are robust to urban and metropolitan influences, but whether our waterways have the right food resources for them. So they're eating mostly like shellfish. They're mostly carnivorous. They're eating, yabbies and shellfish and other things like that. And whether those are persisting with all the carp in our water bodies and all the effluent and everything else that goes in them, I don't know. But another place where we need a lot more research to understand what's happening to them.
Fingers crossed, we might just have to look a bit harder and get more citizen scientists sending in their records whenever they can. There's another question here from Jackie. We'll just have a few more, and then we better let Kevin have his evening back. But what can we do within our means to provide suitable habitats to protect both insect species and mice and rodents in terms of planting native grasses, plants, trees, etc., in our own backyards?
Well, I'm not really an expert on habitat restoration, but I think the idea of planting native plants and gardens and planting gardens in general, it will be a big help to many species, certainly insects and flowering plants. Whether you'll get native rats in your garden, depending on how suburban you live, I don't know. I haven't had rats except black rats return to my garden yet, and I've done a lot of planting. But I think, in peri-urban and suburban gardens and suburban habitat plays a really big role in preserving biodiversity. One of my colleagues works in the Wimmera. He works for the Wimmera CMA, and he was saying that the tree cover in Melbourne is greater than many parts of the Wimmera because of the row crops. So, good on Melbourne for doing its part to create habitat, right? We have a huge gray-headed flying fox colony because of many of the trees and plants we've planted. They have just returned, I guess, to the Botanic Gardens again, where they're becoming a bit of a nuisance again. They were moved from there to the Yarra River site, but they've found the Botanic Garden again. I was just there this weekend seeing the havoc they're causing. But I think planting will do a lot for, and insects. It's a lot of, I assume insecticide use is a big problem. So try to limit the amount that you're using insecticides. I know we have pests we're trying to get rid of, but most insecticides kill all insects, not just the pests.
Yeah, and moving away from that sort of the lawn into more messy, I guess, gardens that have a bit more diversity. There's lots of studies that have shown that insect diversity grows exponentially moving away from lawn to even some native grasses. Also having a little bit of areas of mulch and things where insects can actually breed. And also keeping your, a lot of those eucalypts with the sort of hanging bark. A lot of people sort of get rid of that ribbony bark because it looks unattractive, but it's a haven for a lot of insects. So things like that as well. There's a question here about telling the difference between a black rat and a swamp rat. Rattus lutriolus.
They're saying they think they have swamp rats. So that's exciting.
Got any key features to look for, Kevin? If you're just seeing it on a real quick glance.
The key feature is the tail. I mean, for me, the face looks different. We have, some of us can recognize faces, but if you're just getting them, so black rats have really long tails. Black rats are also called roof rats because they're arboreal and they live in our roofs. They climb. It's one of the ways you can tell it's a black rat versus a sewer rat or a brown rat. Is black rats will climb up, and most of our native rats can climb, but they won't seek shelter that way. And then also swamp rats are really, kind of dark. They have dark feet. They have really dark feet, and they have a kind of really chestnutty kind of golden hue to them, and are much darker. They have smaller ears. They look just ever so slightly less ratty. So like a black rat has big ears. He looks kind of pointy nosed and has usually lighter feet, but black rats can be all kinds of color. It's kind of a misnomer. They're called black rats, but they're usually kind of a gray brown rat, and they have very long tails that are longer than their body.
And might be a little bit biased, but swamp rats are also like they're really, really cute. They're super cute. They've got chubby little faces. They're real cute.
Yeah, they're definitely more blunt faced and puppy dog than a black rat.
That'll probably do for questions, but there was someone adding in that they used to see Ricali in Greensboro on the Plenty River in the 70s and 80s growing up there and walking around. So yeah, that's promising that they used to be there. So we just need to keep an eye out, I suppose. But thanks so much for giving us a tiny insight into all your expertise. That was awesome. I learned heaps. I hope other people did as well. Thanks so much for your time and presenting.
Was also interested in knowing more about sort of creating habitat for sort of native rats and for insects and things in their gardens or bush blocks or wherever. The Gardens for Wildlife program is a good starting point for that. It's a community program that where volunteers offer come and offer some advice on creating habitat for wildlife. We can send that link around maybe after the webinar.
Exactly, and keep following along with some of our e-news for other events and other talks, other expert walks and talks, and yeah, things like this. We're trying to host as many as we can. So reach out if you have any ideas, and yeah, make sure you all thank Kevin for his time.
Thank you all. Thank you for your attention.
Thank you so much.